Hi everyone, our paper studied the telomerase of tetrahymena to discover that it was a ribonucleoprotein enzyme with two kinds of primer specificity.
During the time that the article was written, the importance of ribonucleoproteins or RNPs as an essential component for various biological processes was becoming increasingly apparent. These structures, which contain both an RNA and protein components, have been historically identified as useful for ribosomal RNA processing, protein synthesis, mRNA processing, and much more. This article focuses on the addition of telomeric repeats at the end of nuclear chromosomes as an RNP-facilitated process. In eukaryotes, linear chromosomes risk losing genetic material during each round of replication due to the fact that there is no more sequence for an RNA primer to fit into, so that DNA polymerase is capable of replicating the ends. However, telomerase, also known as telomere terminal transferase, functions to combat this issue. The focus for the remainder of the article will be on the organism Tetrahymena, although it is important to note that telomeric sequences are conserved across many organisms. Many organisms' telomeric sequences, similar to the image on the left, have GNC repeats and balance the loss of genetic information with the addition of extra sequences by telomerase. Tetrahymena cell extracts are used because they were previously discovered to have shown the ability to add telomeric repeats in a non-templated manner using only a single-stranded telomeric sequence oligonucleotide and DGTP plus DTTP. Thus, scientists proposed two mechanisms that allow for the incredible specificity displayed by telomerase enzyme activity. These include the recognition of the G-rich oligonucleotides both structurally and sequentially. Previous research had revealed that in vitro conditions, there were specific single-stranded oligonucleotides that were able to prime telomere repeats TT, GG, GG in both tetrahymena, the organism being studied in this paper, as well as in yeast, which is one of biology's most studied model organisms. Taking this one step further, they tested various oligonucleotides that corresponded to G-rich telomeric sequences of different organisms to further understand the primer requirements for the elongation reaction. To conduct the experiment, telomerase was purified over a gel filtration column in which the column was calibrated to identify the region where the telomerase was expected to elute and run on a 60% polyacrylamide 7 molar urea sequencing gel with a resolution of 1 base. Each primer was also 5N labeled with 32P to act as a molecular weight marker. They were able to determine that the G-strand oligonucleotides from tetrahymena, seen in lanes 2 through 4, Saccharomyces, seen in lane 5, Trypanosoma, seen in lane 6, Oxytrichus, seen in lane 7 and 10, and Dictylostylium, seen in lanes 15 and 16, prime the addition of tandem repeats of the 6-base tetrahymena telomeric sequence. The only difference with these lanes in 15 and 16 is that the telomere was additionally purified on a heparose column. Additionally, they found C-rich oligonucleotides did not prime elongation, along with the control that contained no oligonucleotide primer, seen in 13. Furthermore, they wanted to test the ability of telomerase to recognize tetrahymena telomeric sequences of varying lengths. The sequences they tested are shown in lanes 1 through 4. They found that the hexamer D, TT, GG, GG did not prime elongation, while multiples of this repeat did with increasing efficiency. In previous experiments, they determined that the distinct 6-base banding pattern was due to pausing or chain termination before T residues. This information allowed them to visualize when the TT, GG, GG sequence was added onto the 3' end of the primer and conclude that sequence addition is correctly initiated at different 3' ends. When testing the 24-base yeast oligonucleotides, they observed a 1-base upward shift in the repeat pattern than when they used the 24-base tetrahymena oligonucleotide. It is important to note that the yeast oligonucleotide has a T followed by 3 Gs versus the tetrahymena, which has a T followed by 4 Gs, both on the 3' end. From this observation, they tested to see whether this shift came from the 3' end of the yeast primer or from some other aspect of the oligonucleotide. Tetrahymena sequence oligonucleotides with 3 or 4 Gs at the 3' end were used to prime elongation, causing an upward shift by 1 base, seen in lanes 3 and 4, and 4 bases in lanes 3 and 5, which confirmed the belief that extra Gs are added onto the 3' end of the primer to complete the first T, GG, GG sequence before additional TT, GG, GG repeats are added. Lastly, they found that putting an A in the sequence is not recognized as equivalent to the original G residue in the sequence.
The purpose of the gel filtration of telomerase is to determine the molecular weight of telomerase. There are two different types of gel filtrations that have to be carried out in order to fulfill this purpose. These gel filtrations are the analytical gel filtration and the preparative gel filtration. The goal of the analytical gel filtration is to determine the presence and relative proportions of substances in a mixture. whereas the goal of the preparative gel filtration is to isolate and purify a sufficient quantity of a particular substance in a mixture. In the preparative gel filtration experiment, researchers used polyacrylamide beads, and the region where telomerase was expected to elute was marked. Tetrahymena S100 extracts were loaded onto the column. 1 liter of flow-through was obtained, then all 302 milliliter fractions were collected. Every 10th fraction was tested for telomerase activity, and all 300 fractions have been stored and frozen in liquid nitrogen after they have been collected. In the analytical gel filtration experiment, researchers used agarose beads, and the molecular weight of the standards eluted from the column was verified by running protein gels on the peak fractions. 1 milliliter of S100 tetrahymena extracts were loaded onto the column. Fractions were collected, and every other fraction was tested for telomerase activity. Due to unsuccessful attempts to determine the molecular weight of telomerase and no recovered telomerase activity, researchers couldn't determine the specific molecular weight value of telomerase. Instead, they analyzed the active fractions, which are highlighted in green in the figure on the right, and found the molecular weight to be between 200 to 500 kilodaltons. Researchers had problems with stabilizing the purified telomerase enzyme, which can explain the issues they had with determining the molecular weight of telomerase.
In general, for nuclease digestion, a couple experiments were performed with MNase and RNase A alongside active fractions of telomerase. Figure 4a shows how the addition of MNase, which is a micrococcal nuclease, or RNase A, affects the overall activity of telomerase. We see that increased concentrations of MNase and RNase decrease activity. Oxidized RNase A and RNase A plus RNasin, which is an RNase inhibitor, and MNase with EGTA, which is a chelating agent that binds to calcium and inactivates MNase, end up having no effect on telomerase activity. The main conclusion is that it is the nuclease activity of MNase and RNase A, and not simply the physical presence of these proteins, that inactivates the telomerase. In figure 4b, we see different levels of purified telomerase, 2 columns of purification versus 4 columns, that are pretreated with RNase A at increasing concentrations. Some lanes have RNasin added before the RNase A pretreatment, which overall inhibits the RNase A. Some lanes don't have RNasin at all. We mainly see that RNase sensitivity isn't limited to crude telomerase fractions from gel filtration columns, and applies to pure fractions too. The overall trend is seen in both conditions, but more so in the higher purified telomerase fractions. In general, figure 4a and 4b show that RNA and protein components are necessary for telomerase activity. Figure 5 looks to see whether telomerase elongation can be regained. We first treat active telomerase with or without MNase. Then we inactivate MNase with EGTA, which is a chelating agent, and add different RNA preparations to both the nuclease-treated and non-treated fractions. In general, we see that activity isn't restored at all.
Due to the large size of telomerase, along with its protease sensitivity and requirement for specific RNA, it was suggested that telomerase was a ribonucleoprotein. In order to determine this, they needed to identify the RNA species that co-purified with telomerase activity. To do so, an extensive purification scheme was completed. The column series was a preparative gel filtration, heparin agarose, diethylaminoethyl agarose, or DEAE agarose, cybercron blue agarose, and spermine agarose. These purification steps employed the ion exchange chromatography method. Once these steps were completed, the columns were phenyl extracted and the RNA was end-labeled onto a 7-molar urea sequencing 6% polyacrylamide gel that contained DNA molecular weight markers. Looking at the gel-labeled activity, we see that the telomerase activity eluted off the spermine agarose column in fractions 10 and 12. Looking at the gel-labeled RNA, we see that most of the RNAs eluted in the later fractions 22 through 26. In order for the DNA to co-purify with the enzyme activity, the RNA would have had to elute in a similar fraction as the telomerase. They believe that this discrepancy in eluting fractions comes from the fact that the telomerase is inactive at salt concentrations above 500 millimolar. This means that the lack of enzyme activity in the higher fractions comes from the inhibitory effects of high salt concentrations. From these experiments, they concluded that the most likely candidates for RNAs of telomerase were approximately 154 and 80 nucleotides in length. This is because the RNA species that ran near the DNA markers of 154 and 75 were always found in the active fractions. In conclusion, this experiment told us that the small RNAs of 150 and 80 nucleotides in length co-purify with telomerase.
This article was groundbreaking in determining that telomere terminal transferase was responsible for both recognizing the G-rich strand of DNA and adding telomeric repeats. Using sequencing gels, they concluded that the RNA component was responsible for recognizing the G-rich strands and adding bases accordingly. The molecular size of telomerase was determined by gel filtration and found to range from 200 to 500 kilodaltons. The large size is an indication that telomerase is an RNP with an enzymatic function. The RNA itself was confirmed to be crucial for telomere addition when RNase inhibited function. Specifically, RNA acts as a template for repeat addition. In this image, we can see the mechanism of telomeric addition. The G-rich strand of DNA is longer, leaving a single strand overhang for which the RNA component telomerase acts as a template during replication. The telomerase also additionally acts as a reverse transcriptase, serving as an active site for RNA-dependent DNA synthesis. The complementary C-rich strand can be synthesized by DNA polymerase once extension has occurred.
The specificity components in telomerase were found to be similar to those of tRNA nucleotidal transferase, an enzyme that is able to recognize the structure of different tRNAs in order to add a 3' CCA sequence. However, the synthesis of this sequence is not dependent on a DNA or RNA template. The article also speculated about the RNA component's possible involvement in determining the sequence of the telomeric repeats that are synthesized and/or specific primer recognition. Later research cloned the RNA component and found that it contained a sequence that provides a template for the synthesis of T2G4 repeats. This template is an integral component of telomerase and makes it unique compared to other eukaryotic RNPs in that it uses an RNA template for DNA synthesis, which they theorize may come from an ancient process. Furthermore, while the article found that RNAs of telomeres about 154 and 80 nucleotides in length co-purified with telomeres, further research needs to be done to completely rule out the possible presence of other RNAs that may be undetected by the RNA labeling experiments done in this article.